All right, welcome. Um, Today's video is we are going to read through 3.1 up to mitosis because I know that if I say, hey guys, take out your books and read this on your own, it's not going to happen. You'll get your books out, you'll glance at it. It's fine. So I'm going to make you this Ed puzzle where I'm going to read through the section and then ask you questions along the way. So you are forced to basically read it with me. All right. So we are on page 84 in our books, but I'm going to zoom in here. 3.1 titled The Cell Cycle and Cell Division. Okay, so if we take a look at our book here, we got our vocab terms highlighted in yellow, and here we go. The cell cycle. No matter where you live, you have probably noticed that the weather changes in a regular pattern every year. Some areas experience 4 seasons: winter, spring, summer, and fall. In other parts of the world, there are only 2 seasons: rainy and dry. As seasons change, temperature, precipitation, and the number of hours of sunlight vary in a regular cycle. These changes can affect the cell cycles of organisms such as trees. Notice how the tree in Figure 1 changes with the seasons? Like change in seasons or the growth of trees, cells go through cycles. Most cells in an organism go through a cycle of growth, development, and division called the cell cycle. Through the cell cycle, organisms grow, develop, replace old or damaged cells, and produce new cells. Phases of the cell cycle. There are 2 main phases in the cell cycle: interphase and the mitotic phase. Okay, so they try to label in this figure down here interphase in different shades of blue. So all 3 of these blue slices are interphase, and then in yellow is the mitotic phase. Interphase is the period during the cell cycle of a cell's growth and development. A cell spends most of its life in interphase, as shown in Figure 2. During interphase, most cells go through 3 stages: rapid growth and replication, or copying of the membrane-bound structures called organelles, copying of DNA, the genetic material in a cell, and preparation for cell division. Interphase is followed by a shorter period of the cell cycle known as the mitotic phase. A cell reproduces during this phase. The mitotic phase has 2 stages, as illustrated in Figure 2. The nucleus divides in the first stage, and the cell's fluid, called the cytoplasm, divides in the second stage. The mitotic phase creates 2 new identical cells. At the end of this phase, the original cell no longer exists. So here's interphase, where we go through the rapid growth and replication, copying of the DNA, the genetic material of the cell, and then the preparation for cell division. And then after that, we go through the mitotic phase, which is significantly shorter than interphase. And in the first part, mitosis, the nucleus will divide, and then the cytoplasm, cytokinesis, will divide. And when that cell splits, you end up with 2 cells that are identical. Identical means they are the same. They're basically clones of each other. And you might ask, hey, what happened to the original cell? It no longer exists, okay? So that one cell now becomes 2, and you get 2 new identical cells. The original cell is no longer there, okay? Length of a cell cycle. The time it takes a cell to complete the cell cycle depends on the type of cell that is divided. Recall that a eukaryotic cell has membrane-bound organelles, including a nucleus. For some eukaryotic cells, the cell cycle might last only 8 minutes. For other cells, the cell cycle might take as long as 1 year. Most divided human cells normally complete the cell cycle in about 24 hours. As illustrated in Figure 3, the cells of some organisms divide very quickly. So Figure 3 up here, it says the fertilized egg of a sea star divides into 4 cells in about 2 hours. It divides into 1,000 cells in about 10 hours. Moving on to interphase. As you have read, interphase makes up most of the cell cycle. Newly produced cells begin interphase with a period of rapid growth. The cell gets bigger. This is followed by cellular activities, such as making proteins. Next, actively dividing cells make copies of their DNA and prepare for cell division. During interphase, the DNA is called chromatin. Chromatin is long, thin strands of DNA, as shown in Figure 4. Let's take a look at Figure 4. During interphase, the nuclei of the animal cell and of a plant cell, contain long, thin strands of DNA called chromatin. When scientists dye a cell in the interphase, the nucleus looks like a plate of spaghetti. This is because the nucleus contains many strands of chromatin tangled together. All right, phases of interphase. Scientists divide interphase into 3 stages, as shown in Table 1. So here's interphase, and we can divide it into 3 stages, G1, S, G2. And then we have a description for each one of those stages. Interphase begins with a period of rapid growth, the G1 stage. This stage lasts longer than other stages of the cell cycle. During G1, a cell grows and carries out its normal cell functions. For example, during G1, cells that line your stomach make enzymes that help digest your food. Although most cells continue the cell cycle, some cells stop the cell cycle at this point. For example, mature nerve cells in your brain remain in G1 and do not divide again. So there are some cells in our body that, once they are made, they no longer go through cell division, and neurons is one of them. And that is why when people damage their nerve cord, their spinal cord, and they basically cut the connection between those neurons there, they're paralyzed. They can't use a part of their body. And neurons, they don't divide again. They don't repair that connection. So take care of them. During the second stage of interphase, the S stage, a cell continues to grow and copies its DNA. There are now identical strands of DNA. These identical strands of DNA ensure that each new strand gets a copy of the original cell's genetic material. Each strand of DNA coils up and forms a chromosome. Identical chromosomes join together. The cell's DNA is now arranged as pairs of identical chromosomes. Each pair is called a duplicated chromosome. So this is called a duplicated chromosome. 2 identical chromosomes called sister chromatids. So here's one sister chromatid. Here's the other sister chromatid. Make up a duplicated chromosome, as shown in Figure 5. Notice that the sister chromatids are held together by a structure called the centromere. The final stage of interphase, the G2 stage, is another period of growth and the final preparation for the mitotic phase. A cell uses energy, copying DNA during the S stage. During G2, the cell stores energy that will be used during the mitotic phase of the cell cycle. All right, organelle replication. During cell division, the organelles in a cell are distributed between the 2 new cells. That means that they get split up evenly between the 2 cells. So here we have 2 cells that are just about ready to split, and they have divided up their DNA and their organelles and other contents evenly. Before a cell divides, it makes a copy of each organelle. This enables that the 2 new cells function properly. Some organelles, such as the energy process in mitochondria and chloroplasts, have their own DNA. These organelles can make copies of themselves on their own, as shown in Figure 6. Oh, I guess this is a mitochondria. But if these were 2 cells going through cell division, they would have split their contents evenly. A cell produces other organelles from materials such as proteins and lipids. A cell makes these materials using the information contained in the DNA inside the nucleus. Organelles are copied during all stages of interphase. All right, moving on to the mitotic phase. The mitotic phase of the cell cycle follows interphase. It contains of 2 stages, mitosis and cytokinesis. In mitosis, the nucleus and its contents divide. In cytokinesis, the cytoplasm and its contents divide. Daughter cells are the 2 new cells that result from mitosis and cytokinesis. During mitosis, the contents of the nucleus divide, forming 2 identical nuclei. The sister chromatids of the duplicated chromosomes separate from each other. This gives each daughter cell the same genetic information. For example, a cell that has 10 duplicated chromosomes actually has 20 chromatids. All right, so let's just talk about that for a second. Here I have a duplicated chromosome. 1 duplicated chromosome. How many sister chromatids do I have? 2. 1 sister chromatid, 2 sister chromatid. Imagine that I had 2 of these, 2 duplicated chromosomes. So one right here, and we'll just say 1 side by side. How many sister chromatids would I have now? Would I have 1, 2, 3, 4? I'd have 4 sister chromatids. Okay. When the cell divides, each daughter cell will have 10 different chromatids. Chromatids are now called chromosomes. The definition of a chromosome, fun fact, is counting the centromeres. So you basically count the centromeres, whatever number of centromeres you have, that's how many chromosomes you have. In cytokinesis, the cytoplasm divides and forms the two new daughter cells. Organelles that were made during interphase are divided between the daughter cells. All right, I think this is where I want to stop. Yep, this is where I'm going to stop.